well. I hope you can stay with us throughout the COVID series I've put together, and I'd like to take this opportunity to thank everyone's understanding as well in moving the original World Vaccine Congress dates from this week. And a big thank you to our webinar sponsor, TriLink Biotechnologies, for supporting and helping us deliver these as well. As you may know, at the same time tomorrow and the day after, there will be 2 more COVID sessions. But today, I have the pleasure of introducing our moderator, which is Dr. Philip Corresi, Deputy Director, Office of Vaccines Research and Review at CBER at the FDA. He oversees activities associated with regulatory review, which includes all vaccines against infectious diseases. More recently, he's also been contributing as the ad hoc advisor to WHO as the chair of the COVID-19 vaccine working group for the R&D Blueprint team on topics ranging from the clinical trial design and analysis, vaccine stability, as well as development of the research roadmap for vaccines against SARS-CoV-2, which is, of course, what has all brought us together today. Just a note as well, just so you're all aware, you can submit questions using the control panel. Phil will bring some of these up during the Q&As after the presentation. And our speaker today, as you're all very familiar with, is Dr. Gregory Poland, who's chair of the Congress Scientific Advisory Board. Him and many of our speakers are incredibly active in interviews recently, so I'm sure you've seen a number of these already. But that's enough from me. Over to you, Phil.
Yes, well, thank you very much. I'd like to welcome you all. The topic today certainly doesn't need any introduction. In fact, I struggled quite a while to find a single recent article in the newspaper on any other topic. But what's needed is clear information and thinking, and there are few better people more capable of leading a discussion of this topic than today's speaker. He's an expert in COVID-19, and incidentally, he has a podcast which very amply demonstrates his ability to bridge the gap between science and public communication. So if you like what you hear from today, you might check that out at the Mayo Clinic website. Dr. Gregory Poland is the director of the Mayo Clinic's Vaccine Research Group. Which is a state-of-the-art research group and laboratory that investigates issues surrounding vaccine response and novel vaccines important to public health. He's the director of the Immunization Clinic and director of the program in Translational Immunovirology and Biodefense at the Mayo Clinic, and also serves as the president of the Edward Jenner Vaccine Society and is the editor and the chief for the journal Vaccine, and is, of course, a full professor at Mayo in several different departments. So without further ado, Greg, we'd like to hear what you have to say on the topic of SARS-CoV-2 going viral.
Well, thank you, Phil, and thank you, Wing. Thank you to all of you that have joined us. Of course, different circumstances than we expected, but we're adaptable, and we hope more adaptable than viruses. So with that, we will get started. I want to, in our time today, describe what we know about this novel coronavirus, talk a bit about infection of humans, and then some of the control methods that are being used through public health, antiviral, and of course, discuss vaccine approaches. I'll start with Albert Camus, who won the Nobel Prize in Literature, writing about a plague in a different era. And he said they fancied themselves free. And no one will ever be free so long as there are pestilences. So it'd be interesting if we were there in person. I'd ask how many of you have been infected with coronaviruses, and hopefully you would all raise your hands. And that's because there are 4 known seasonal beta human coronaviruses. You see them listed there. Those are endemic. They circulate every fall, winter, account for about 30% of seasonal upper respiratory infections, and in otherwise normal hosts are generally benign. Human coronaviruses were not really known about until the 1960s, even though in animals they were known in the late 30s. Just a very brief review. This, of course, is the 3rd novel coronavirus in the last 18 years. SARS-CoV, as it's now known, in 2002 in Guangdong province in China. About 8,000 known infections. There were super spreaders known. It had about a 10% case fatality rate, and it used the ACE2 receptor. That will turn out to be important as we talk further. MERS, and I should say, by the way, that SARS spontaneously disappeared in July of '03. MERS, we have not been as lucky. This has a much higher case fatality rate, about 35%. Again, super spreaders have been identified. This used a different receptor, the DPP4 receptor, and was a bat coronavirus with the intermediary host of camels, still small numbers of cases in the Arabian Peninsula. Well, George Bernard Shaw said that we learn from history that men never learn anything from history. I included this because it is remarkable that here we are with the third introduction with no antivirals that have been licensed, no vaccines that have been licensed. And I think it relates, interestingly enough, to the executive summary of the 9/11 Commission, where they said, we believe the fundamental problem was one of imaginability. We could not imagine it, and therefore we failed to prepare for it. The other side of the coin, as my wife said, he did say men, not women. Okay, SARS-CoV-2. This was first identified in December of 2019 in Wuhan, as everybody knows, with cases probably first happening in the October-November timeframe. No one really knows. Corona is just a Latin term for the crown-like appearance of the virus because of the spike proteins that you see there. Well, this turns out to be very important. So as we talk, you'll hear me refer to the S protein or the spike protein, but note that there are other important structural and non-structural proteins that also offer antiviral and vaccine approaches. Now, what's interesting about coronaviruses is the really extensive ecologic diversity, primarily in bats, where over 500 different coronaviruses have been identified. The other interesting thing about it, and not surprising as an RNA virus, it does accumulate mutations, but it is thought to do so at the rate of about 2 a month or so, to put a time frame on it. And for that reason, you'll notice that, for example, Netherlands 63 virus up at the top, they figure that that emerged in the 1100s, which is sort of interesting. So you can track that. It is a single-stranded positive RNA virus. I mentioned the mutation rate. A group of investigators at UCSF have put out some data. I haven't seen a peer-reviewed publication yet on 8 different strain types that they figure have occurred in the amount of viral mutation that's occurred thus far. Certainly mutations have already been detected in the S gene. Their significance is unknown, but potentially would have implications for vaccine development, particularly with what I call S-only approaches. Oh, sorry, okay. One of the things that I'm often asked to speculate about, and I choose that word carefully, speculate, is will we see seasonality with this novel coronavirus? And the problem is nobody knows. I've tried to tell the media and others that if the canvas that we call SARS-CoV-2 was blank 13 weeks ago. And it really is a testament to science that we have filled in so many pixels on that canvas already. But it also means there's still a lot unknown. We certainly do know that there are seasonal human beta coronaviruses, as we talked about. SARS disappeared in the summer of '03, but there is a large pool of susceptibles. And cases have already occurred in the warm season, if you will, in Australia, Africa, and the Middle East. There will undoubtedly be movement now into the southern hemisphere as they move into their fall and winter. And the question will be, will that blow back to the northern hemisphere this coming fall? Again, no one knows. So how does the virus infect humans? There are multiple methods. Of course, inhalation of viral-laden larger respiratory droplets, aerosolized virus, touching contaminated surfaces, And like SARS, I think we have to entertain the possibility of an oral fecal route. Certainly, virus is shed in stool. That's been identified. There is a clinical corresponding phenotype with diarrhea, abdominal pain, and nausea. So how big of an issue that is as yet unknown. I want to point out a paper that appeared, a week ago or so in JAMA, because I think they do a good job of indicating that emission clouds from sneezes, coughing, talking, probably cigarette exhalation, etc., are much more complex than the simplistic binary way we've tended to look at it as aerosolized or large respiratory droplets. And their point, which I indicated down there, was the gas load and its payload of pathogen-bearing droplets of all size probably travels out to more like 27 feet. The other thing to think about is in public places, washrooms are actually a very dangerous place to go. Obviously, when you got to go, but not only are you crowding hundreds to thousands of people in the smallest room, say in an airport, for example, but most public toilets nowadays don't have covers. And so when you flush a toilet, there is a cloud plume that results. This has been elegantly demonstrated for other viruses, for example, norovirus, showing that it very effectively covers every surface in the bathroom with detectable virus. Similarly with forced air hand dryers. Well, SARS-CoV-2 has been, of course, detected in respiratory secretions, both nasopharyngeal and oropharyngeal, blood, stool, and the environmental surfaces in rooms of infected persons. I should add one other thing, the outside of masks that we wear. This cartoon, I think, is important. I won't spend a long time on it, but I do, as we talk about the S protein and some of the approaches that are being taken, I did want to give you a visual of this. The receptor, as we mentioned, is ACE2. It has a high density in the lower respiratory tract, but it's also present in colon, bladder, and other human tissues. The S protein is actually a trimer S1 head and the S2 component with a membrane-bearing portion. That turns out to be important as again we talk about approaches. Now, I thought this would be interesting to show you. These are similarities in gray and differences in red with SARS S protein compared to SARS-CoV-2. And you see that while there's a lot of similarity, there are also differences. And those differences can relate to how we think about developing antivirals and vaccines and how they might work compared to those that were developed for SARS. Now, one of the questions I often get is why lung tropism as sort of the hallmark, if you will, of this disease. Well, just to show you two quick studies to give you a sense of it. One, a study of normal lung tissue among normal donors. 83% of the ACE2 expressing cells are alveolar epithelial type 2 cells. That's important because they're particularly dense in the lower respiratory tract and serve as a reservoir for viral invasion. When you look at gene ontology enrichment analyses, what's important about alveolar epithelial type 2 cells is that they have very high levels of viral process related genes. Again, so that's a favorable environment, if you will, for the virus and facilitates coronaviral replication in the lung. Now, this leads to a number of approaches that we can think of. If you look to the far left of the screen, of course, that spike protein is a prime target for development of a vaccine, also for the development of soluble ACE2 to bind up that spike protein. One of the serine proteases that activates the S protein is called TMPRSS2. and that's known to have several different isoforms. Their significance not clear with this disease, but developing TMPRSS2 inhibitors is another approach, as well as ACE2 receptor blockers. So clinical features, I think these are all very well known to anybody listening to this talk, but I will say that we need to add to this probable cardiovascular disease phenotypes, including myocarditis, arrhythmias, evidences of vascular inflammation, and neurologic, as has been seen in other viruses. Some of you may have seen this very nicely written report of I think about a 40-some-odd-year-old woman who developed acute hemorrhagic necrotizing encephalopathy as a result of COVID-19. This diagram, which appeared early on in the New England Journal, I think is a very important one to keep in mind, because it relates to so many of the questions about COVID-19. One needs to realize that because of the relative lack of testing, what we see is the tip of the clinical pyramid. That is, those both sick enough to seek care and having the ability to seek that care, and then that care being able to offer testing for diagnosis. How deep and how wide the base of that pyramid is yet to be seen and unknown, although that will quickly change as serologic assays come online. One of the, of course, important issues regarding the transmissibility of this virus is the reproductive number, which has been variously estimated somewhere in that, just to use round numbers, 1.5 to 2.5 range, and certainly does account for how this disease has spread. To put that into perspective, a different virus that my lab has studied for over 35 years is measles virus, with an R0 of in the 12 to 18 range. So again, just to put into perspective that reproductive number. Well, some significant clinical issues include the variable incubation period, varying between 3 and at the outside, and the outside is more modeling, but 28 days with a mean of about 5.5 days. Certainly, asymptomatic transmission has become obvious. Prolonged shedding, there have been a number of case reports now in regards to that. To my knowledge, true super spreaders have not yet been identified. It's become clear that not only lower but upper respiratory symptoms and GI symptoms occur, that there is a diversity of the spectrum of disease presentation, often centering around factors such as age and comorbidities, but we'll talk about others too. And the other interesting thing is pulmonary abnormalities seen on imaging studies, even in otherwise asymptomatic patients. Now, this is a graphic from the Lancet that was based on an analysis of 41 patients. And this is pretty much held true in terms of from the time of onset of symptoms to admission to the hospital. Our own experience at Mayo Clinic has been this is pretty accurate, with shortness of breath developing very quickly thereafter. If they're going to move on into ARDS, that follows on along with concomitant admission to an ICU. I would say what is somewhat different is the gender disparity has not been quite as dramatic as this, which was a function of low numbers. Nonetheless, there continues to be, whether we look at very symptomatic people such as adults, or some of you may have seen the MMWR. Actually, I saw the preprint. I'm not sure if it's been released already, but even in children, the gender disparity continues to hold up. Mortality rate variable by risk factors, et cetera. So this incubation period that I mentioned, about almost 98% of those who are going to develop symptoms seem to do so within about 11 days of infection. But note that the 95% confidence interval goes out to almost 16 days. Keep that in mind as we talk about the quarantine times of 14 days. And then Lauer et al. Had a very nice paper looking at this, and under conservative assumptions, about 100 out of every 10,000 cases would develop symptoms after 14 days. So again, that's important in thinking about public health strategies. Further, when you look at the proportion of symptomatic infections that have yet to develop symptoms, you'll notice that that 99th percentile extends out to and past 28 days. So will that become important? I think so. And I think it plays into, sort of when do we begin unwinding this or declare all clear? I personally think that what we're going to have to do, and it will be a hard ask of the economy and the public, is that once we see a significant diminution in cases or even no cases, we'll need to wait somewhere perhaps as long as 28 days after that, taking into account that lag period. All right, in terms of disease severity, I won't read this to you. These are relatively large series here. This first one, almost 45,000 people. This would be consistent with what we are seeing. Notice the very high case fatality rates, though, in Wuhan, on the bottom point compared to the rest of China, and we'll talk about that in just a second. When you look at case fatality rates, one of the things that's important, and I think has escaped a lot of attention, is that that case fatality rate very much depends on the surge demand on the healthcare system. So this is not variations or mutations in the virus. This is not expertise in training in treating cases. This is not the availability of new therapies. This is stage of the epidemic. So when you flood the hospital system, the mortality rate goes up and then it drops as you have lesser cases. So this is an important strategy in the so-called flattening the curve. The other thing, of course, well known now, is that the case fatality rate does vary by, and this is over 72,000 cases, again, all China here, but by comorbidities. I'll point out that hypertension, that one initially, and in fact, we published a letter about it, made a number of us concerned about ARBs as an example. That does not seem to be holding up, and hypertension is probably more of a marker for cardiovascular risk rather than hypertension treatment per se. The other thing I think well known now has been the case fatality rate by age. So when we talk about overall rates, which is important at one level, I think we also have to be much more individualized, as we are in the rest of medicine now, to say, well, these can vary strikingly by age and comorbidity. Laboratory findings, just to very briefly describe them: lymphopenia, probably most often seen, but you see the range there. Elevated aminotransferase levels. Interestingly, the observation that often on initial admission, serum procalcitonin levels have been normal, but then become elevated with more severe disease. Recovery time, I think, is still a bit of an open question, but notice that for severe disease, it's been as long as 6 weeks. I think we'll probably find out a range larger than that as time goes on. You've seen, I think, some of these chest x-rays. Notably, you tend not to see pleural effusions. You tend to see bilateral rather than unilateral involvement, and lower lung involvement more than upper. Ground glass opacifications and consolidative abnormalities are extremely common. Less common, as I said, are effusions, pleural thickening, or lymphadenopathy. The most severe lung abnormalities tend to be seen about 10 days after symptom onset. Again, that would agree with our own experience. And as I mentioned, CT abnormalities seen prior to symptoms and even prior to detection of viral RNA. Now, I do think that some of these oddities that we have seen deserve more careful scrutiny, both in the operating characteristics of the tests that might have been used during a tsunami of cases in China, and what other concomitant respiratory viruses may have been circulating. And I think you've seen some of these CT imaging too, showing basically the same elements of consolidation and ground glass opacities, particularly in the lower images. In terms of antibody and immunity, we have no known correlate of protection at the Mayo Clinic. Yesterday, we started doing serology. For seasonal human coronaviruses, I'll point out that immunity tends to last in the range of months. With SARS, immunity, two different studies that I thought were decent studies was shown in lasting about 2 to 3 years in 90% of recovered studies in one study. And in another study by 6 years, SARS-specific IgG antibodies and peripheral memory B cells were undetectable, although they were able to identify some T cell anamnestic responses. So the answer to how long will antibody last with COVID-19 in terms of protective antibody, no one knows. Anybody who answers that question with a number is speculating. Clinical management, I think, again, is becoming increasingly clarified. One point that I think worth making is that large doses of corticosteroids, absent some other indication in both SARS and MERS, were unhelpful. One of the things that has surprised me as I've talked with clinicians around the world is the relatively simplistic and binary thinking of SARS-CoV-2 positive or negative, and then not looking past that. But I want to make the point that we do have antivirals for other viruses, and I think we're going to find that co-infections are more common than we think, particularly with influenza, and deserve to be identified and treated. I talked about steroids, no reason to go on about that. That's available in the CDC interim clinical guidance. Co-infection issues that I mentioned, depending on where you live, I'll take the US. We have SARS-CoV-2, influenza A and B, measles, mumps, pertussis, RSV, human metapneumovirus, parainfluenza, and a variety of other respiratory viruses, rhinovirus, for example. So the bottom line, I think, here is that you have to have a high clinical suspicion, and you have to do a respiratory pathogen panel to know, are you only seeing SARS, or are there co-infections that deserve to be identified, may alter the management and clinical course, including the use of antivirals. So what about protecting ourselves? Well, the biggest risks are not using PPE, followed by not using it properly, followed by inadequate hand washing, fatigue, and mistakes. And my daughter, who's a mental health professional, has started to talk about the second wave of the pandemic, which are mental health issues. So does wearing a standard face mask work? And I'm going to answer that yes. While there are some contradictory studies, I would say, yes, it does seem to help protect others if the infected patient is masked. I think standard masks do protect against large respiratory droplets, and probably equally as important as a behavioral or memory aid to try to prevent finger to nose, eyes, and mouth contact. Of course, standard masks do nothing against aerosolized virus. For that, obviously, you need an N95 mask. I did want to point out, when you look at the filtering efficiency of the N95s, they're called N95 because they're about 95% efficient. Diameter of a coronavirus is about 0.12 microns. Cough droplets can vary quite widely, but let's just say roughly on average about 100 microns, and I'm really simplifying there. And surgical masks filter to maybe 2 to 10 microns at best, if used under optimal conditions and not wet. This is why healthcare providers get infected. This man has a beard. It's a little hard for you to see in this picture, but he has an air leak around his chin. He's got crowning of the mask above his nose. He's not wearing goggles or a safety mask, and the patients are not adequately separated from one another. So just a reminder about the things we know that do work. I think because they're not high tech, people tend to give short shrift to hand washing, but this is exceedingly effective and underused. Now, the problem is, other than surgeons and infection control professionals, I have never seen anybody, physicians included, none of them wash their hands properly. And the problem is they think they know how to do it. So if you want to learn how, you can go onto YouTube, search Dr. Pollan and Jimmy Kimmel, and we kind of use an innovative way to show people how to wash their hands. Now, why is soap and water better than hand sanitizer? It's because soap products actually integrate into the viral lipid membrane and destroy the virus. They also decrease the electrostatic attraction of virus to hand and hand oils and washes the virus away. Hand sanitizers don't do that. You have to use a hand sanitizer that's alcohol-based. Doesn't work well if the hands are visibly soiled or have mucus on them. So that's why I call it second best to hand washing. Interestingly enough, very early on, when you look into the transmission dynamics and kinetics of this virus, I wrote an editorial which took a couple of weeks, so it's not such new news now, but a couple of weeks to get published on this whole idea of how we have to basically hit, I called it a suspend mode, in order to flatten this epidemic curve. So for those of you that are interested, I give an example in there, and it's at the Lancet ID. Now, this I call my most important slide because I think we have seen the importance of this play out on every national stage and have gotten to watch the natural experiments. So where you, and the hard part for politicians and others to understand is this 14 to 28 day lag period. So once you hit the pause button, it will take 14 to 28 days before you see that curve start to flatten. Some countries have seen that happen, and inappropriately, Japan would be a good example, say, well, cases are down, let the kids back into school, and almost precisely 14 plus days later, a major spike in cases. So this is going to be important in sustaining this long enough to actually dampen and at some point eliminate further cases. Now, we stand in the US in the black there at a very important decision. Countries that decided to do this and did it well, like South Korea, Singapore, I would say not only social distancing, but mask wearing countries, flattened that curve very ably, not perfectly, you can't. But in countries that have decided not to do that, we've seen exponential rises in the number of cases. This is a more complex slide, but I think better illustrates what's happened in a number of the Asian countries. Now, Japan, as I mentioned, went back to business usual too quickly, and now they're joining the left side of the curve compared to countries that did not hit the pause button early and are not enforcing social distancing, mask wearing, etc. And I realize mask wearing is controversial. You may have seen some of the CDC worst case projections. I don't happen to share these. I don't think we're going to see death rates in the US of 200,000. I personally will be surprised if we get into the 50,000 plus or minus 25,000 range. I think these need to be adjusted. Antivirals and vaccines, as you know, none are licensed currently. Either licensed antivirals or candidate antivirals are being studied. I'm gratified to see that more and more countries are insisting that they be used under research protocol. I think it's ludicrous to use these without collecting data unless you have literally no other option. But you're familiar with all of these, and I won't go through the long list of them. Vaccines, none are licensed. The interesting thing will be the regulatory pathway if outbreaks stop. There are methods to do that, and Phil may actually comment on that later. I've listed a variety of approaches that are being taken there. I personally think, and I'm just getting ready to publish an editorial on this, that you may remember that we were hearing things like we'll have a vaccine in a few months, and then it was a handful of months, and now it's 18 months. I think that's very optimistic, and I have concerns that I'll share in a moment about that. These are the sort of major approaches that are being tested. As you know, we're in week 3 or 4 or something like that of the Moderna NIH mRNA vaccine trial, but there are others there. We're taking in my own lab, a pathogen-derived HLA, naturally processed and presented peptides that we will isolate and then package with an adjuvant as a peptide-derived vaccine. But lots of approaches being taken, some 35 to 40 that I've been able to identify. Some concerns and cautions. This is my personal opinion. I have concerns about S-only vaccines. These are, after all, RNA viruses. Mutations in the S protein have already begun to be identified, though I grant we don't yet know their significance. For the SARS candidate vaccines, there was pulmonary immunopathology seen and a hepatitis-like picture seen both in mice and ferrets after viral challenge. I personally think, and obviously you can't develop them and license them all at the same time, but I think, and we've argued for the idea of a more individualized vaccinology approach like we're doing in medicine. I think we're going to need more than one vaccine type for the immunosenescent elderly, for the pregnant woman, for the immunocompromised, and maybe even immuno-immature children that are at risk. Just to give a timeline that I think is pretty optimistic, but the cost to develop vaccines is considerable. And CEPI has been brave enough to put out a timeline and a cost, which I think can at least inform the discussion about when these will happen and maybe something a little more realistic. I personally think these are vaccines that we'll see in late 2021, early 2022, if everything goes perfectly. So caution, immense public and governmental pressure for treatments of all sorts and vaccines, but there is no substitute for careful clinical studies and trials. And I am worried that we're rushing. I think we have to remember that case reports are anecdotes, and the plural of anecdote, the old saw goes, is not data. They merely provide hypotheses to be tested. And we must always remember as physicians, the oath we took: first, do no harm. So summary, facts and not fears. The majority are going to have no or very mild symptoms, and the more significant morbidity and mortality is principally among adults and those with other comorbidities. I think we should talk to people in terms of engaging in what I've called contextually appropriate layers of protection, which starts with suspending non-essential activities to basically suppress that R naught to 1 or less. And then I think one thing we have learned, and I have railed about this in innumerable talks and innumerable publications over my 40-year career, is that we have to have better imaginability. We have to be better prepared. This is the third one in 18 years. We could have, should have been better prepared in our testing, in antivirals, and even in later stage vaccine development. So I will end the way I began with Albert Camus. Everybody knows that pestilences have a way of recurring in the world, and yet somehow we find it hard to believe in ones that crash down on our heads from a blue sky. There have been as many plagues as wars in history, yet always plagues and wars seem to take people equally by surprise. So I will stop there, and Phil, turn it over to you for moderating any questions and comments.
Well, sure, Greg. Excellent presentation. You covered a wide landscape. And of course, one of the #1 topics of articles and editorials in the newspapers these days is the role of hydroxychloroquine in treating people who have SARS-CoV-2 infection. So could you say a few words about if and when or under what circumstances you would consider using that drug, and anything else you'd like to say about it?
Yeah, and I'll comment broader than just hydroxychloroquine. I mean, certainly those are available to be used. It is speculation as to their efficacy or effectiveness, I should say, based on laboratory studies, to my knowledge. And I think that what they provide are hypotheses to be tested. So I have no problem with using them at a variety of clinical stages of severity. The idea of saving all of these therapies for very, very late cases strikes me a bit like the person spraying water on the barn that's already been burned down. I think we need to know at earlier stages and in people who have risk factors for progression to more severe disease, but to study them, whether it's plasma-derived therapies, antivirals, to study them under protocol where we can quickly garner data. For a number of these antivirals, I understood that the French consortium was to release early results Friday. I have looked and have not found those results, but I expect that we'll probably see some this week.
Thank you. So another question related to response. And of course, we look at different responses in different states, some of greater versus lesser severity. But then we also look at least what we saw in February is the response that the Chinese were putting into place, at least in Wuhan and perhaps other places, which seemed rather extreme, in some cases locking people in apartments, or at least one was reading that, and perhaps more stringent quarantines than people are being asked to undertake here in the U.S. So the question is, do you think that the response is adequate to really get this under control, or will a more severe response such as that China put into place be necessary to really control this in the U.S.?
Yeah, it's a very good question, and you know what I wish I could show would be quantitative data to say, you know, go to this point and no further, but nobody has that. What we can say is that social distancing works. And so within the limits of the law in an open and not totalitarian society, how do you best do that? Particularly when you have a differential severity, let's call it, by age, and a differential imaginability of how bad this can and could be. And I'm talking particularly about spring breakers, younger people, et cetera. I personally think that hitting the pause button is appropriate. I think we pushed it late and should have learned quicker based on what we were seeing in China. I think our quarantine times and the way we handled things like cruise ships, even what's happening in the military right now, are inadequate, slow, and deserve to be better informed. It is a interesting thing to observe the interplay between science, politics, and economics. And not always the right thing gets done, in my opinion, from the scientific point of view because of that interplay.
Thanks, Greg. So along those same lines, for instance, in many areas, restaurants, while they're not open for sit-down traffic, do have takeout windows. And yet, of course, the food in restaurants is prepared by people, some of whom might be asymptomatically infected. Should all restaurants be shut down?
Yeah, I really don't know the answer to that question, to be fair. I do share concerns that when you're talking generally, I'm generalizing here, about food preparers, they are often the least educated, often poorly trained entry-level workers who cannot afford to take a day off. I've talked with a number of them. They will come to work sick because they cannot afford to not get that pay, sometimes each day or by the week, in cash. So I harbor reservations about this. At the same time, we don't have any clear data saying that, okay, say somebody who was asymptomatic or pre-symptomatic touching a styrofoam container leads to cases. Now, the absence of evidence doesn't mean that it couldn't be true, but it's really hard to thread that needle.
Got it. What can you tell us about the role of testing, whether it's ultimately serological or expanding the availability of, for instance, PCR or nucleic acid testing for the presence of virus in getting this under control? What do you think needs to be done there?
Yeah, that's a really good question, Phil, and I think deserves careful thought as part of a roadmap for how we proceed from here. I think both are necessary. I think we need, ideally, if we can get there, point of care diagnostic assays, because it does matter, even in the absence of specific treatments. If I knew that I had it and I had somebody in my family who had a risk factor for more severe disease, I would do things differently than if I didn't know that I had it, for example. The second is the serologic assays, as you talked about. I think these are going to be really important in terms of the unwind. How can we deploy people back into the working sector, the teaching sector, the healthcare sector, et cetera? One way would be to understand who's been previously infected. Ideally, we would at some point be able to point to a correlate of protection with those antibody tests. But I think the combination of those and the widespread, easy availability of those is really important in how we proceed from this point forward.
Thanks, Greg. There's a question here about vaccine enhancement, and as that is a theoretical risk of vaccines and perhaps proceeding too hastily. And yet, of course, balancing the desire to get a vaccine quickly. You want to say a few words about that?
Yeah, I am concerned about this because of the probable antibody enhancement that has been seen in other failed vaccine attempts, was seen in the preclinical work with some of the SARS vaccine candidates. You know, you're talking about a number of mechanisms where you can have non or sub-neutralizing antibodies and binding to a virus, or complement dependent mediated mechanisms, or as was seen in some of the pulmonary immunopathology in mice and ferrets, a direction of or a bias of the immune response toward either Th1 or Th2, or at least an imbalance, or increased viral replication because you've suppressed an antiviral response. I think, and original antigenic sin as yet another mechanism. I think all of those deserve to be investigated, deserve to be thought about, and plans for detecting that, and I know those are present, but plans for detecting that explicitly in vaccine development is important. One of the things that alarms me a little bit, and Phil, you've got more expertise in the regulatory side of this than I do, but I'm alarmed a little bit of the concomitant non-human primate and human studies in phase 1 vaccines, rather than a small animal model, a non-human primate, gather that data, scrutinizing it, and moving on to humans. And granted, there's a tightrope we're walking here, but in a generally vaccine skeptical society where influenza has killed far more people so far this season in the US, and yet we have at least moderately effective vaccines, that we can't get 60% or more of the highest risk groups to use. All we need is a bad side effect from a vaccine being deployed too quickly. And I think we'll have real issues on our hands going forward.
Yeah, I guess since you called me out here, Greg, what I can tell you is that regulators are very carefully considering these issues and are looking, of course, at a lot of data that they view as addressing these issues adequately, at least in the context of the vaccines that have gone forward. But they are looking at each vaccine on a case-by-case basis to make sure that sufficient data exists to assure us that the kind of bad outcome you're talking about doesn't happen. Go ahead.
We have an excellent regulatory system, in the U.S., but it's notable that people will complain that it's slow, and I would say it's designed to be, in the sense of vaccine development needs to be reflective, it needs to be peer-reviewed, it needs to be evidence-based, so that, as you're saying, we proceed in a safe fashion, which we are.
Great, thanks, Greg. So there are several questions about antivirals, and I might not have caught up to all of them, but one question about azithromycin, another one about convalescent plasma and perhaps monoclonal antibodies, and another one about are there really early phase, highly investigational antivirals that may have some chance of coming online to help with this?
Yeah. So my own view of this is the most immediate thing we can do is plasma-derived therapies. Those have been used way back in the great influenza pandemic of 1918. For those that are interested, Thomas Luke, L-U-K-E, published a review of that in the Annals of Internal Medicine, I think around 2010 or so. Showing the efficacy even then, before we knew much of the science. So I think that's going to turn out to be an important therapy and a bridge therapy until we can get through these clinical trials at least early clinical trials of the wide spectrum of antivirals and antibiotics that are being considered. You raised the question about azithromycin. I don't know of any evidence that azithromycin by itself has any antiviral effect, but rather its effect may be either in combination with another drug or specifically because of bacterial co-infection.
Okay, another very interesting question. There was an article in the New York Times a few days ago about studies of using BCG, the TB vaccine, to hope for off-target effects on immune responses that might help with this. And in fact, there may be other strategies involving other vaccines that may induce general type 1 interferon responses that might play or could theoretically play some role in reducing susceptibility to this infection. What's your take on that? Of course, some clinical trials have started, but what are your thoughts?
Yeah, it's an interesting idea. Vis-a-vis BCG specifically, I have concerns about widespread administration of that drug. There are, and well-known, some significant side effects to that. I cannot imagine that its value would be anything more than an indirect sort of general, if I can simplify it this way, revving up of the immune system. It's not even clear to me, though, when one would do that. Would you really want to stress the immune system, hoping for off-target advantages, and at what level of COVID-19 disease severity? I think any idea is worth potentially testing in a clinical trial, but again, I think there's way too much enthusiasm about it as opposed to thinking about it as a testable hypothesis.
Thank you. Do you have an idea of how much greater the risk currently is for healthcare workers than average people in the community? Or are there any assessments of the current risk of infection or bad outcomes for healthcare workers?
Yeah, I don't think the outcome is necessarily different for healthcare workers, other than we tend to have, in the US I'm speaking of now, a generally older healthcare workforce and a generally unhealthy society in terms of risk factors. And I think we're seeing that in the US. I think Italy has clearly seen it. I think they have something like 90 physicians that have died now. Something in that order. I think issues of PPE availability, proper use have been issues. Obviously, healthcare workers in certain settings are exposed to very high inoculums. And I think disease severity might logically vary by inoculum size. So I think a lot to learn there.
Sounds good. We're close to the top of the hour, and I want to be sure to put in a plug for the talks that will be taking place later this week, tomorrow and on Thursday at the same time at 11 o'clock. And in fact, there was a question about One Health and the role of animals in the spread of disease. And maybe that's one that we can take tomorrow when Dr. Mark Carley, who's the Center Director of the National Animal Disease Center, is going to be talking on the topic of coronaviruses in livestock, vaccines, and international transport of virus. And of course, on Thursday, we'll have Barney Graham, who's the Deputy Director of the VRC and Vaccine Research Center at NIH, and he's been intimately involved in the development of one of the vaccines that's currently in clinical trials. And so he will likely have some very interesting things to say as well. So I hope those of you who are signing off now at noon will join us for those talks as well. There's one final very interesting question here, which sounds like it's from somebody who has a specific question for you, Greg. And that's whether the Mayo Clinic has patient samples that can be shared for epitope analysis of immune responses in recovered patients. And so maybe if you could address this general question of how people are sharing samples and how well the research community is coming together to make sure that we actually come to the kinds of answers that we need as quickly as possible.
Yeah, one of the things that's interesting to me, and again, this canvas was blank 13 weeks ago. And it's astounding how the resources of science and medicine have gathered together to produce a lot of knowledge, but there's still competition. People have their own samples from their own institutions, and I know even for us within Mayo Clinic, the samples are not abundant enough that all of the proposed studies, even within the Mayo Clinic, can get done. And I think that's generally the case for a lot of institutions. I think data is being shared. I think collaborations are being rapidly formed. But we really do need, particularly, for example, the antivirals and vaccines, we need large collaborations across medical centers. Mayo Clinic was designated Friday as the National Coordinating Center for plasma-derived therapies. So that may be one avenue there.
Thanks, Greg. And what I can say from my involvement with WHO efforts is that there is a highly coordinated international effort to bring people together, to share data, to help design experiments, and to really try to come to an answer. And specifically in the vaccine arena, I think that nobody wants to be in a position where we get a vaccine at some point, however many months or years from now it is, and we look back and say we could have gotten there quicker or more efficiently. And so really no effort is being spared to get us where we need to go. And yet I think your point is well taken, is there maybe aren't enough samples. Do you think it's also because people are concentrating more on doing research than on actually getting samples, which may be less glamorous, but ultimately may be equally important?
Yeah, I mean, I think there certainly are elements of that. The institutions that have the most samples are literally trying to keep their head above water, much less trying to figure out, all of the IRB and other MTAs, et cetera, that have to happen in order to share samples.
And so one other question here that I hadn't gotten to, since we're afternoon, we still have over 200 on the line. The question about fecal-oral spread. Do we really know that the virus survives in stool and can come out in the feces? Or could this be viral RNA, for instance, from virus that has found its way through the alimentary tract, starting obviously in the mouth? How certain are we that fecal-oral is a significant mode of spread for this virus?
Yeah, so we don't know that, as I mentioned. We do know that for SARS. And so there's the precedent to be thinking about this. The studies I'm familiar with thus far are RT-PCR assays. So they merely tell us that genetic material is there. There is one study on a preprint server, so take it for what it is, of being able to culture virus out of stool, and for as long as, I think it was 30 days. So that needs to be confirmed and sort of stand by until there's more research.
Okay, somebody asked, and Wing can address this when we're done, whether these slides will be posted or can be shared. And another late question here of whether you want to comment on cytokine storm or the role of cytokines in clinical disease.
Oh, that's a very good question. And actually, I cut out some of those slides in the interest of time, but I think there is the clinical sense that one of the things that we're seeing in very rapid deteriorations, and this occurred to a friend of mine who's a physician, where undoubtedly a very rapid rise in fever, severe myalgias and arthralgias that required narcotics to deal with, and changes in important vital signs and volume status, et cetera, are undoubtedly cytokine or chemokine driven. This has led, in a case of a couple of studies, to begin to look at cytokine inhibitors as a potential life-sustaining therapy. I think it's real. I'm speculating based, as I say, on clinical cases, but definitely deserves to be investigated. It's a very good thought.
And one final question before we close then, and this is sort of a vaccine question, and this has to do with, it's drawn from several questions that came in, and that is, given the fairly short time frame of protection with most coronaviruses, and even relatively short with SARS, and of course, maybe there are differences in how hard people looked, but what do you think is actually a realistic duration of protection for a vaccine against this disease?
Yeah, it's a great question. At one level, you could think of a vaccine whose immunity would last a year plus. And the idea behind that strategy would be to immediately suppress any disease, i.e., if transmission is no longer occurring, the disease is not a threat. Now, maybe you'd have to give boosters or something. The ideal, of course, would be a 1 or 2 dose series, or perhaps it would have to be periodic, that would give long-lasting, ideally lifelong immunity. But I think we're way past what we actually know about durability of protective immune responses. This, again, is another issue I have with S-only vaccines and humoral immunity-driven vaccines only, rather than T-cell memory approaches. I think we'll develop something, of course, and we need that immediate need, and then we'll get more sophisticated in 2nd and 3rd generation vaccines.
Fantastic, Greg. So Wing has asked me to let you know that the webinar is recorded and will be shared with everyone. So you will be able to get the recording, which will be emailed to you in an MP4 format. Are there any final closing remarks you would like to make, Greg, before we finish up?
Yeah, I maybe, at the risk of maybe being too emotional, but this has been a long, hard slog for everybody. I know Phil and I were talking, everybody's working 16-hour days for the last 12 plus weeks. It is remarkable what together we have accomplished. And I would just urge people, don't think of yourself as isolated in your work. Please do collaborate. Please don't fatigue. We need high quality information as quickly as we can get it, and be a voice of appropriate skepticism and reason. I really think that it's going to be a process to rein in politicians, the public, and the media toward a more science-based approach, even in the midst of what seems to us unusual and novel. But recall that we are the first generation, as far as I can tell from history, that has not faced plagues and wars and famine and floods as nations. So this is not novel. People have come through this before with far less resources. We have, through the new biology, unbelievable speed and kinetics around the world in attacking this. So just urge everybody, thank you for what everybody's doing. Do be collaborative, and let's work hard to learn a lot of science that we can apply to preserve health in people's lives. So thank you for the opportunity.
Fantastic, Greg. Thank you for an excellent presentation and very thoughtful answers to these questions, and also a downright inspiring closing statement. So for all of you who've attended, we all greatly appreciate your attention, and we all, I think, have work to do now in whatever our capacities are to get through this. And so we will close now, but I do look forward to noticing many of you logging on to the subsequent sessions later this week, tomorrow and Thursday, both at 11 o'clock. So thank you again,
And
Greg.
Phil, if I one part I neglected to say, specifically thank you to you and the FDA, who must be going 1,000 miles an hour trying to work with all the investigators that are trying to get INDs and EUAs, et cetera. So if you could relay our thanks back to the agency.
Absolutely. There's no question that there are a lot of people who are putting a lot of work into this and certainly doing their very best to deal with what is an unusual situation for everybody, to say the least. So I will relay that. Thanks a lot, Greg.
Thank you.
So thanks to our listeners. Wing, did you want to make any final comments?
I think you both put it so eloquently that I wouldn't do it justice. But yeah, I mean, thank you to you both so much. Thank you to all our listeners. And as we've been saying, 2 more exciting sessions the following next 2 days at 11. So please join us then.
Okay, goodbye.
Goodbye, everybody. Thank you so much for your attention.